So now 2023 paper, first question. Option A. Yes, option A is the correct one. Again, this question is based on the structural organization from the animals. So different kinds of epithelial tissues. So like the chart we have seen for the connective tissue for 22 set B question, this kind of a epithelial tissue chart will be help you to understand means their functionality, then appearance that they have, and the location where they are and what kind of a structure they have. So squamous epithelial tissue serves as a diffusion barrier for different organs and the different body parts. Cuboidal epithelial have they have their nucleus at the center of the cells and also helps for the secretion and absorption. Columnar epithelial have their nucleus at the basal part of the cell and they are also responsible for helping for the secretion and absorption. And ciliated epithelial have the nucleus at the basal side and also helps for the movement of the particular particles. If you remember the ciliary movements that we even talk about when we talk about the fallopian tube. So in the bronchia also for the mucal and the particular particles movement, this kind of a tissue, this kind of a cells are also present. So option A is the correct one. Next. This part is also deleted from your syllabus. Digestive system, I don't know why, but they have deleted this particular chapter from your this year's syllabus. Sorry? Yeah. Yes. Okay. So you can, if they have already given you the answers, then you can challenge it. That's why they have given the answers. If you are very sure that the other options are already there, if you are agreed, no problem. But that kind of a pattern is you can see for thread 22B that we have solved just now. They have also given answers for 22B. Okay. Yeah. So you can see means this is your for your practice. Once you are get convinced that yes, option A is the correct one, then no problem at all. Hmm. 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 Okay, 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 okay. So let's see. We will solve the paper, and if we see that all answers are real, then no problem at all. Okay. So this part is deleted from your this year's syllabus. Digestive system part they have already removed. So if you have any answers for this question, hello, am I audible now? Okay. Okay. So, as this part is deleted, I'm just giving you the answer. So, peptones are partially digested proteins. That is the correct one. Option A is the correct one. So, these are the partial degradation will be happening at each process, and finally, in the small intestine, amino acid will be the smaller units will be the basic unit from the peptones will be formed and get absorbed eventually. So, option A will be the correct one for question 2. Next. Okay. Yes, option A is the correct one. So biomolecules and their corresponding nature is what we are talking. So this is from the biomolecules. So insulin is example of the protein. This is an enzyme which is a proteinaceous in nature. And as you all are aware, all the proteins are the heteropolymers. They are not homopolymers, which is a characteristic of the all the proteins. Then inulin is a kind of a carbohydrate, it's a polysaccharide. So polysaccharides, the characteristic that you can correlate is the homopolymer that they have. Then lectin is a example of the secondary metabolites produced by the different kinds of a plant, and lecithin is a quaternary derivative of the ammonium. Next. Mm, Yes, yes, correct. So in the metaphase, the spindle fibers, the aster fibers will be, which are the kinds of a microtubules will be formed. So that will be eventually getting, if the drug is acting on that particular part, then the metaphase will be the first phase in the karyokinesis where it can get affected very easily. So option A is the correct one. Next. Anyone? So this question is based on the plant physiology part. So we have to find out the incorrect statement for the seed structure. So firstly, you need to find out the seed structural part. So I have included this particular diagram. So monocot seed and dicot seed, they have different kinds of a layer, different kinds of a structure that they have. So if you can remember this particular structure, then we can answer this very easily. So the first statement is the monocot seeds, the membranous seed coat that is fused with fruit wall is called the alluron layer. No, that is the incorrect statement, because if you can see in this particular diagram, the seed coat and the fruit wall is already there, and the alluron layer is the inner layer which is present in between the seed coat and the endosperm. So that is a separate layer. It is not, once it is fruit with the fruit wall, it's not called the alluron layer. Alluron layer is a different layer itself. So the first statement is the incorrect one. The endosperm is not present in some of the mature dicot seeds. That is true, because endosperm is a nutritional layer which is consumed by the embryo, developing embryo, for the many dicot plants. So for the fruit development, so that is used for the energy source. So that is the correct statement. In dicot, the outer layer of the seed coat is called testa. That is the correct statement. And coleoptile and coelurhiza is found in the monocotyledon seed is again the correct statements. Next. I think this part is also cut from your syllabus now. Yes, this part is also, this is from the plant growth and development, but this part is entirely cut, right? This part is, yes, and not entire thing, but this part is particularly cut from this year's syllabus. So I'm just answering this particular part. So you have to understand for this particular thing, two things are there. If you remember, there are rings developed. If you cut the wood in between, the tree trunk is cut, you can see rings in between, which are responsible for estimating age of the plant. So for this, various kinds of factors are there. One is called late wood and early wood. So early wood is what we call the spring wood, which is formed during the spring season. And then there will be the autumn wood, which will be found in the late season. So two kinds of things that we can see based on the seasonal part, the rings will be helpful to form, which will have the different kinds of a vascular system in between them. And then there will be the part we call the sapwood and heartwood, which will be the also one. It's for the development that happen in the particular season. That's what we are referring it with a different name. And hardwood and softwoods were help for developing that kind of a rings that can help over the development. So outer part that we can see the rings that will be seen in the autumn wood, and the inner part which is helpful in between the two rings is the part which is developed in the spring region, which is the spring wood. And the heartwood is the innermost core part which is present in the wood, which usually have the dead kind of a tissue. And the sapwoods are the one, and it's more over the hard kind of a wood, which is usually used in the material for the furniture pipe because it's hard in nature. And softwood can also use for, especially for the pulp region, because it's still have the conductive tissue. And the name itself is a sap itself, which is a soft in nature. So that's why it is a softwood or sapwood. However, we can see the outer rings, means as the age increases, we can see the sapwood will be present on the outer side. So here we are referring to the temperate climate, which is a uh, colder than the, which is present in between the polar and the tropical region. So in the temperate climate region, the it's moreover the autumn wood and the sap wood which is responsible for the estimating the correct age. So by which we can see the ring structure more clearly. For this particular question, option D is the correct one. So if you are confirmed that this your answer is correct, and though the once they release the key or the paper, and if you can see option A that they have given for the all the question, you can challenge at that time. You can to the IISER, you can challenge this kind of question that I have this kind of a data. So I'm proposing for this particular question option, the another option which is the correct one than the option that they have given. Yes, yes, yes. Next. A or sorry, B. Okay. So let's see each option. We have to find out the correct statement for biological nitrogen fixation. So catalytical redox center of the nitrogenous contain molybdenum and iron as a cofactor, which is the correct statement. It requires these two metal ions as a cofactor for the functionality. Atmospheric nitrogen is fixed in the nitrogen is by converting nitrogen to nitrite. No, firstly the atmospheric nitrogen, its conversion happening by the process of the ammonification. So ammonification is the first process, and then by nitrification process the nitrate formation will be happening. Then nitrogenase can function optimally only in the presence of the molecular oxygen. No, nitrogenase can function under the anaerobic condition. So anaerobic condition is what is required for its functionality. And the transport of the importance of the amide produced by the transamination to different parts of the plant body is mediated by phloem. No, it is mediated by the xylem. So xylem is responsible for the conduction of these particular amides which are produced. So B, C, D are the incorrect and option A is the correct one. Next. Yes, option A is the correct one because here the example that we are referring is talking about the genetic diversity. So we have to first find out where the genetic diversity for the particular statement is applicable. So if let's assume the A is the correct one, that because now you are all aware that A is the correct answer that they have given. B is the higher diversity of amphibians in the Western Ghat than the Eastern Ghat. This is the comparison between the geographical diversification, two different geographies that we are referring. So it's an example of the geographical diversification. Great variations of the ecosystem found in India than the Scandinavia. Again, the two different geographies that we are referring to. Then the greater diversity of the plant species found in India as compared to Central Asia. So here everybody is talking about either species diversity or the geographical diversity, which is more focusing. Variations in the potency of the concentration of reserpine produced by the particular kind of a variety that we are talking about is because of the production of that particular metabolite product is because of the genetic diversity that can cause the variation among that particular species. So that way option A will be the correct one. Next. Anyone? Mm. B. Okay. So if you remember the process of the transcription translation, the two things are different, right? So formation of the mRNA will the process of the transcription. where the rho factor and sigma factors are there. Sigma factors is involved in the initiation of the transcription, means at the early transcription process, and the rho factor is at the release factor for the completion of the transcription. So these are the two ones. Option B and D are involved with the process of the transcription, and here we want to focus on the process of the translation. So we can easily eliminate B and D. Release factor is the correct answer. Means option A is the correct answer. There are, especially in the eukaryotic system, there are release factors which are present in the ribosome itself. Once the ribosome started reading the different kinds of a codon, once they reach to the termination codon, these release factors helps to bind with the stop codon, and the signal is given to the ribosomes that the protein synthesis should be stopped at this particular point, because stop codon is already there, and no more tRNA will be recruited at that time for the elongation of the polypeptide. So ERF1 and ERF2 are the two kinds of release factors which are associated with the these are two releasing factors which are associated with the eukaryotic translation process means stopping process, termination process. Next. Is my screen visible? I'm sorry. Yes, option A is the correct one. So here we are talking about the different systems which are associated with reproductive system. So Sertoli cells are the one which provide nutrition to the developing spermatozoa. Follicular growth hormone is associated with the triggeration of the ovulation. Then the placenta is the one which secretes the gonadotropin hormone, and urethra is the one which carries the urine away from the bladder. Next. Anyone? Mm. Anyone? Anaga, Shreyas, Kirti. Am I audible? Okay. Okay. So two parts we have to refer for this. So we are talking about a causative agent for the different diseases. We have to consider the examples of given for the different diseases. And here we are talking about the causative agent which requires splicing of their hnRNA, that is heterogeneous nuclear RNA, to generate the mature mRNA. So first understanding that we can have the splicing of the hnRNA to mature RNA. This process is involved in the case of the eukaryotes. So if the eukaryote system that we are talking about, so we have to find the disease where the eukaryotic causative agent is involved. So we have examples of the malaria, pertussis, typhoid and tuberculosis. So if you can see, pertussis, typhoid and tuberculosis are caused by bacteria, different kinds of a bacteria. Means pertussis by Bordetella pertussis, then typhoid by the Salmonella typhi, tuberculosis by Mycobacterium. These are all the examples of the bacteria. So if they are bacterial origin, the splicing process will not be the case for the development of the mature RNA formation for them. This for the in and the other hand, malaria, the causative agent of malaria is the Plasmodium. Plasmodium is the parasite which is a eukaryotic in nature. So this is a because of that eukaryotic nature, the splicing process is involved in the causative agent for that particular disease. So malaria will be the correct answer. Next. Yes. So we have to find out the inhibitors and the combination with their particular substrate combinations that they have. So if you remember the concept of the inhibitors have the similarity to attachment with the active site that they have. So here the enzyme that we have and the active site, as you are seeing, it is a triangular in nature. So the competitive inhibitor have their structural similarity with the substrate. So that way they can compete with the active site of that particular enzyme, and because of the structural similarity they can get attached and the substrate attachment will be unavailable for that particular enzyme. So by understanding this, the substrate is a triangular in nature, so we can see the P and S have this kind of these two potential inhibitors have this kind of a structures which can work as a which can get easily attached with the active site. So option A is the correct one. Next. Anyone had solved? Yeah. Yes, option A will be the correct one. So Hardy-Weinberg equilibrium, again, you can see this question many times. This is a very favorite part for IISER aptitude test. So we know the equation P square plus 2PQ plus Q square is equal to 1 and P plus Q is equal to 1. So we have to find out the heterozygous combination. So we are looking for this particular value. So we have only 4 individuals out of 10,000. So we can find out the value of the Q. So by q will be the square root of the q square value that we have got from this particular thing, because only 4 individuals amongst 10,000 are positive for the Zika, and this is the recessive disorder. So we have, we can, once we get the q, that is 0.02, we can easily get value of the p will be 0.98, which is the dominant allele present in the population. So calculate the heterozygous population frequency. We will calculate the value of the 2PQ, which will be 0.0392. So this is a frequency. Now we want the number of individuals. So once we get the frequency multiplied by number of the population, we get the value of the 392, which will be the heterozygous individuals for this disease of the sickle cell anemia in given population. Next. It's not clear. Scary, it's not. It's just very easy. Just read it carefully. Okay, 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 okay. One of the, whenever you see this kind of error, firstly, don't get scared. That's the one thing. Understand what problem that they have given. That 100 base pair DNA fragment is cloned between the given plasmid. This is the first situation that you should remember. So they have given the insertion site, that this is a region between these two. This 1000 base pair insert is inserted. So the original plasmid size is 3.5 kb. So 1000 is added. So the new plasmid that will be formed by the insertion of the 1000 base will be 4.5 kb. This is one understanding that we have done. Then what they did confirm the, so this is the one of the usual process that we do to confirmation of the particular insert is inserted inside by the molecular way, if we want to decide that whether the given transformant have the particular insert in it. So either we do PCR with the specific primers, and another way is to do the restriction digestion map. So what the condition that they have discussed over here. So they have given the two types of digestion. One is a not one and eco R1. And second set that they have this in plasmid is then means two types of digestions will be done. So it's not together thing that you should understand. So first lane will be the plasmid is first digested with not one and eco R1, and then loaded the DNA sample in the lane one. And second is then the plasmid is digested with not one and hint two hint 3, and the plasmid is then loaded in the lane 2 and the patterns were checked. So let's see if no, and here you can see there are multiple not one sites, even they are present in insert also. So first digestion pattern that we can get with the not one and equal one. So not one will cut over here. So firstly we have to decide how much fragments we will get. That will easily eliminate the many options that they have given. So not one and equal one. So not one first cut will be here, then equal one will be here. So cut the fragment number 1. Then not one is also present over here. So this will be the second fragment. Then not one is also here. So this together will be the third fragment, and the remaining will be the fourth fragment. So by first digestion, we will get 4 different kinds of a fragment. So even looking at the options that you have in the lane 1, where you can find 4 fragments is only option A. All others have no fragments available. They are either 3, 2. That is not the case. So you can easily select that. Let's cross check with lane 2 also. So then the plasmid get digested. Then means the first digestion is not there. The plasmid is separately digested with the not 1 and hint 3. So if this is the case, the first fragment will be the not 1 to not 1 to not 1. So this will be the one fragment. Then not 1 to hint 3. This will be the second fragment. Then this will be the third fragment, and this will be the fourth fragment. If this is the case, then lane 2 will also get the 4 fragments. So instead of looking at the size, you can firstly eliminate that how much fragments are there. Based on that, you can select option very easily. If and when there are two options with the 4 fragments, then you have to look for the size that they have given and then choose very easy. Then you can choose what kind of a sizes are there, and then you can have the answer. Here the elimination is very easy because four fragments are there, and 4 fragments is only option available with option A. So you can choose it very easily. So please don't get scared whenever they give the gel electrophoresis. Just you have to make sure that where is that particular restriction enzyme position is there and how much cuts will be there. Firstly find out that, and then if the similar options are there, then look for the size. Okay. Last question. Hmm. Yes, means this is your understanding. Then based on that, you can choose the answer, right? Hmm. Means we have to find out the genotype. So that's why they have given the question mark. Good. And what will be the genotype? Yes, that is correct one. Because once you can see that is easy way to understand. Firstly, it's not a shaded. We have to find the genotype for the this. It is keeping one generation. So we have seen the dominant characteristic usually have they can pass to every generation, and recessive can skip the generation very easily. Here we are seeing from the affected parent. the generation that we have, the second part that we have given, the uh, unaffected individual is or child is born. So firstly here X-link is not possible. The recessive characteristic will be there, and we are interested in this particular part. So this mating is resulted into one affected and one unaffected one. So that means this condition can only present when the two individuals are heterozygous in nature. Then only the characters can get pass by this particular combination in the recessive trait. So that's why first understanding that you can get very easily that as this is the unshaded part that we can see, this means the individual is not affected one. So that means if it is able to can pass through the disease very easily, it is very easily the heterozygous one. So this is the autosomal recessive heterozygous kind of a individual. So option A will be the correct one. So that is good thinking, Anaga. You can easily choose the answers based on that.